Okay, so to kindly introduce all the steps that can be done to work on zoosporic parasites and mention qPCR as one of them. And that's usually a good choice when you know what you're looking for. So if you want to, just a few contact informations. I have a Twitter account. Simona here has a Twitter account, and our institute has a Twitter account, and you're welcome to use that to say how much you like this lecture, of course. So we'll have a brief look at why Simona and myself are here today, because we don't work on algae. Present the 3 cases that connect us to COST Aqua, and then go through a couple of steps in qPCR, design, evaluation, considerations to take when you embark on qPCR. All the resources that come from our side also for the practicals later today, I put on a Dropbox, which is where you can steal that. And yeah, I actually also put the QR code so you can take a picture now if you want to download those, and maybe sit where you can grab the stuff for the repository from them. So just a brief background. We're from the University of Bern, and we're actually from the veterinarian faculty, although I'm a biologist, and from the Department of Infectious Diseases and Pathobiology. So we approach these from the disease side and from the disease side in vertebrates, and invertebrates as well, actually. But it's stuff you can see without a microscope. And in this department, we're the Institute for Fish and Wildlife Health. So we have a fish division and a wildlife division, and veterinarians and biologists work together there. And at this institute, we do research, and usually at the interface of biomedical stuff, population ecology, veterinary pathology. So it's quite diverse. Teaching also to stakeholders, and diagnostics. And this is really why we are here, because especially the diagnostic units often face also zoosporic disease agents, basically. So I'll tell you now about just highlight 3 ways where we get in touch with these so that we can approach us and ask us later on. One is Simona's project, actually, that's about the crayfish plague. The agent is Aphanomyces astacei. And I know zero usually about the phylogenetics of these things because I'm the molecular person. But crayfish plague, what it does is affects native crayfish. They lose limbs. It grows on the surface, but it also grows inside. The native species, they die, and we have invasive species that are carriers. So this is another layer of diversity, maybe, that these things in some groups of species attack some, but not the others. And that's what Simona is researching on, where they occur, which strains occur, and why they sometimes cause disease and sometimes not. Yeah. Then we have Saskia Keller, who works in the wildlife diagnostics, and she's in touch with this class of organisms through BSAL. I don't even try to pronounce that one. And basically, this is a disease that affects salamanders and causes skin lesions. And since salamanders are already an endangered group of species, these diseases contribute to species decline, which is a reason why we're interested. And here, one question is actually that we're starting to tackle a bit is how can we detect this from environmental samples and not just from the animals, because these are not so easy to find. It's actually a question that Simona is also following up on. And finally, we work a lot with aquaculture in the fish health, and in a completely independent project, we did 16S sequencing for bacteria, but then we also tried metagenomics, so basically sequencing everything that we have in there. And here some fungal taxa start to emerge, and I'm pretty sure there's much more. These are just the top 10 phyla. So we're not planning on going in that direction, but the data's there. And if anybody wants to explore this, you're more than welcome to. So qPCR, what do you already know? I need to know first who I'm talking to before I continue. Get your phones or go to your computers, scan this code and answer the questions. And then we'll see how deep I have to go or how superficial I can stay. Okay, I'll move away from this slide now, if it's okay. Everybody got it? Menti and the code? Okay, let's see. Okay, so I think, yeah, good. So you know what qPCR is. It stands for quantitative PCR, so we already are on the right track here. Oh, somebody, that's nice. So uniform. And then when you can move on to the next answer, if this is possible in the program, or do I have to open this somehow? There's another question. You can't? Hey, then I probably need to do something here. Okay. So the next question I have for you, and it's actually, it should be somewhere here. But you can also go to this www.menti.com and use this code. What is the typical length of a qPCR assay? Between 10 base pairs and 500 base pairs. Who would be confident in this group? It's around 150. That's what you say. What's that something else say? Okay, so at least something of the stuff that I tell you today is worth it, because 250 is already quite long. 250 is at the borderline of not a qPCR assay anymore. You want to be in the range of 80 to 120. Yeah, some are even shorter. Okay, good. So let me take you through a bird's eye view of the process of the points where you have to think when you do a qPCR assay. First step is, of course, sample collection. Then the question is, how do you extract your DNA and why you use a certain approach? And that, again, already starts, we won't talk too much about this, but depending on how harsh you have to lyse your cells, that's a big question. And I actually don't know, but I think, Simone, you have struggled with DNA extractions from your organisms. So if you just use the standard sets of tissue extraction or something that's made for mammals and soft cells, they are not necessarily the best way to get the DNA from something that is encapsulated in a spore, in a cyst or something. Then you have the choice between a couple of different types of assays, and we'll talk about that. Primer design is key, as we've already heard. Which organisms do you want to target? How sure are you that you target everything you want? How sure are you that you do not target anything you don't want? And in parallel, in qPCR, to be quantitative, you need a reference. So you need to have a standard, something that you know the concentration of. And you need to create standard curves. And this is not trivial sometimes. We'll do that in the afternoon, so we'll have some fun with that. And all of those feed into the qPCR that you in the end do. Then you have a time of data analysis, meta-analysis, and these feed back into the process. You should expect to go this circle 1 or 2 times if you adapt something from somebody else, and maybe 3 or 4 times if you design it yourself. So account for some time here. And we will today, oh, great colors. It's a beautiful brick, pink. So we will go into these ones in the lecture and the bright pink ones with Simone in the afternoon. Yeah. So just a tiny bit of sample collection. You can have very different types of samples. Very often we have diseased animals, but there is increased focus on non-invasive samplings, for example, for crayfish plague, brushing the host with a toothbrush, and of course, historical samples as well. These are usually fixed and come with their own challenges, like fragmented DNA. So the longer your qPCR assay is, the less likely you are actually to detect anything from such samples. And what we do a lot and focus on a lot is environmental sampling with water filtered. Yeah, the whole team goes out to the field every year as a team and tries to get as many samples for these kind of questions as possible. Simone often goes out at night to see the crayfish as well as get the water. So yeah, has its own challenges. So first question that you have to think about is, do I go from the organism or the environment? Why is this important for the assay design? In the environment, we have inhibitors. We have decaying leaves, we have organic matter, and this will make our assay less efficient. And we have to be aware of that, and we have to control for that, and we have to counteract that. Assay choice. qPCR is not qPCR. So this is a complicated slide, but what I want you to take away from this is that there is two major types of qPCR approach. One is relative quantification. We just want to know whether something gets more, gets less. That's usually in gene expression studies and very often not in diagnostics or detection. So you heard it, you forget about it, okay? Very often, and this is where we are also in the afternoon, it's absolute quantification, for example, of pathogens or of cells. And that usually, this often targets RNA, this most often targets DNA, the genome. And here actually something to be aware of as well is, depending on the target gene, you might target something that occurs in multiple copies in a host genome. And then you get wrong answers about quantities in the environment. So, especially ribosomal stuff could be amplified. And of course, in many cases, we don't care how much, we just want to know if it's there. And in these cases, we could use normal PCR, but the good part about qPCR is that you have a limit of detection and a limit of quantification. That means for qPCR, you can say whether you can't just say it's not there or it's there, but you can say we can detect so and so many copies, and obviously the sample contains less than that, either none or less. So also, yeah, it's a bit more precise in its message. And I really want, if you feel like this is daunting or you want to get more, actually very good source are often supplier handbooks. They really take good care to explain to you how their products work. And there's awesome YouTube videos on qPCR. So Googling is good in this case. And in the afternoon, also to orient yourselves, we will work with an external calibration curve. We work with probes, not with cytokine, but with probes. We have a standard that is a plasmid DNA, and we use, and some of the stuff that we do in the lab also involves synthetic DNA oligos. So basically, there's lots on the slide that this lecture and the practicals will not cover, and just be aware of the other stuff that's out there. Good. So now I want to touch on the two basic molecular assay types that are out there for qPCR. Who of you is using qPCR, actually? 2, 3, 4. Who of you is using cybergreen-based approaches? Yeah, that's next slide. So there is basically, you can find in the literature, if you look for qPCRs for your stuff, 2 types of assays. This one is less often used in diagnostics, but I want to explain it anyways. Basically, it's a PCR, and the more double-stranded DNA you produce in the PCR, the more of this fluorophore of cybergreens, there's a couple of others, is incorporated. So what you see is basically the amount of double-stranded DNA is rising. So that's one, that's the dye-based. And the other is the probe-based assay. And essentially, it also is a PCR, but you have 2 primers, forward and reverse. And then you also have a probe. And this probe is labeled on both ends. On one end, it has a color, a fluorescent probe. And on the other end, it has a quencher. The quencher kind of shuts down the probe. As long as they're in close proximity, this one can't shine. And when they're incorporated, this binds in the middle of the PCR product. And essentially, the shortest qPCRs are just primer binding site, probe binding site, primer binding site. It doesn't have to be any longer, actually. And when this is incorporated, the polymerase will cut off, when it connects here, it will cut off the fluorescent particle. And this will get to move away from the quencher, and suddenly it can shine. So basically here, what you see is the amount of fluorescent signal that is freed from the probe. Yeah. So now what that means is when we think about the assay choice, we want to have to know whether we want to quantify. What is our template, RNA or DNA? Do we want absolute or relative quantification? Or do we have reasons to choose cybergreen versus probe? And the good part about the probe is it makes your assay much more specific, because it doesn't have to bind just the primers, but the probe has to bind too. So with a probe, you can actually use more generic primers and then go in with a more specific probe. That's useful to, for example, see whether a whole family is there, whether something specific is there. Primer design. Okay, for this, I brought a small exercise, and I ask you to pass these on. Don't look at them yet, but just pass them on. And then we can have a look what we take care of when we look at primers. So these are 3 primers that are in use at our institute. You see, one is Aphanomyces, one is Pisal, and then an avian pox virus. Most of you will not design your own primers, but you will read papers and you will want to know whether these primers are already good, or you have 3 primers from the literature, you have to decide which ones do I want to use. And so I'll go through some quality considerations with you now. One of them is primer length. Good primer length is 20 to 25 nucleotides, then you're usually specific and still don't need crazy temperatures in PCR for melting. So when we look at these primers, they all kind of fit these criteria, 20 to 25, that's good. The next thing is primer melting temperatures. And we had this wonderful introduction into base pairing before, GC base pairs and TA base pairs. And what you need to know, or maybe you saw it, is that T and C have 3 hydrogen bonds with each other, and A and T have 2, which means that GC pairs are harder to break up during melting and also form less easily when you cool down. And there's a basic rule that can be applied to calculate these melting temperatures. Every G gets 4 degrees, every A or T gets 2 degrees. And with this, you can easily calculate roughly what is the melting temperature of your primers. And the annealing temperature is usually 6 to 8 degrees less. If you design them, something around 62 to 64 degrees usually works. That's a ballpark figure that is good. And yeah, this is a very, there's many primer design programs, and while they do a good job in finding places that are kind of specific, it's very important to do these hand checks, because these programs spit out primers that I would never design by hand. Another thing that helps to make a PCR or qPCR more efficient is a GC clamp. Now, a GC clamp is when your primer ends on one or more G's and C's, one or two. And why is that good? If you want the primer to bind and then the polymerase to bind and run off and then polymerase more, what you really need is that the end of the primer binds tightly. What you don't want is that the primer binds, but the end is flopping and loose, because then you won't get amplification that easily. And so from these 3 primers, only one of them actually has one of those. And it's sometimes not possible for specificity reasons, but if you can, try to design them. Sometimes you can just move the primer one nucleotide to one direction and order it again. For example, this could, without this A, but maybe have another base here, then you would have. You want the balanced sequence composition, ATs, GCs, and you want to avoid homopolymers. That is a horrible sequence. You don't want this in a primer. This will create tertiary structures. This will bind to any G homopolymer in the genome. This is just not a very good idea. And many primers that you see in the literature have wildcards. These are wildcards. Y, H, N, many others, the UPAC code, maybe you're familiar. Basically, a Y in a sequence, it could be a C or a T. An H could be an A or a C or a T. And this means that you end up with a primer sequence. You can order them like this. They will be synthesized like this. And you have a primer mix that contains gazillions of different sequences. What this means is that some of the primers will have a T here, an A here, and T here, a T here, and so on. And they will have a low melting temperature. And another primer in the same tube might have a C and a G and a C and a G and so on. And they might have a high melting temperature. So this example of our avian poxvirus primers has more than 10 degrees between the lowest and the highest melting or annealing temperature that you can have. And this will, for example, this favor certain variants, certain genotypes of your pathogen and favor others. And yeah, just be aware of this if you have to use one of those. Exactly, amplicon length below 150 if you can, down to 60 is possible. Because the shorter it is, the less your qPCR will suffer from inefficiency issues. Polymerases fall off. Polymerases incorporate wrong nucleotides and so on. And the shorter the runtime from beginning to end, the less likely you run into these kind of problems. I will not go into the primer design business because this will take an afternoon of workshop. But there's many more questions to consider. Really know your primers. Do not just take somebody's primers, apply them, and never question the assay. So what's the time actually? I don't have my watch. 15 minutes. 15 minutes? Okay. In this case, I brought two exercises, which we will not do now, but we can look at them in the afternoon during the practicals, and I can give you feedback. One is sheet. look at these primers and look at which ones should be redesigned. Which ones are bad, which ones are good. Give us feedback. And the other thing is, I have a little R script that I can share with anyone that is able to disambiguate these kind of primers. So if you get such a primer somewhere, how many sequences would this result in? What would be the melting temperatures? What would be the upper and lower limits, and so on. So these are also in the Dropbox folder. Okay, so primer design, we covered that one. Let's jump into standards. Standards are fun. So a standard will help you to design, to determine the properties of your assay in your system. And you use a serial dilution of this. Standards are very often, you have your template that you got, or you that synthesized, or you got it from a PCR, and it's put into something called the vector, a plasmid, that you cut open, you play Lego with DNA, basically, you close it again. And these ones can be produced at very defined copy numbers by bacteria, and then you can always use this DNA as a standard template. And then you create serial dilutions of this and run your assay on this to understand how it behaves on high concentration, low concentration. And what you want to know for your assay, the ones that raise their hands, I hope you know the linear dynamic range of your assay. I hope you know the limit of quantification, that's the lowest number that this assay is able to detect, and the limit of the quantification, the lowest number, where you can be sure about a quantitative result, and the LOD, the limit of detection, where can you still detect it, but you're not so sure anymore about the quantification. And a word of caution here, as we said, our pathogens are genetically diverse. The standard is usually a single molecule of DNA and is usually representing a single genotype of your organism of interest, which means that if you go out to the wild and you have something that has polymorphisms in the primer binding sites, you will detect them less well than the standard. And you cannot really, except if you do really go for metagenomics, you can't know, basically. So standard curves, primary data. What you get in the qPCR machine, as we said, is this increase of fluorescence for different samples. And here you can see the result of such a dilution series from very high concentrations where the fluorescence comes right up to very low concentrations where it takes many, many cycles to raise the fluorescence above threshold. And I marked with arrows now a couple of things that can be noted in this graph. Any of you know what this axis is? Just shout it out. If you do qPCR and you don't answer now, we have to contact your team lead. What is that axis? Number of cycles is the x-axis. Yes, so this is the number of cycles. This is the threshold line, exactly. That's relative fluorescence, exactly. Why do they all flatten here? This is where they exhaust the reagents. That's the plateau. That's called the plateau. When there's not enough primers or nucleotides anymore, they can't, or not enough probe, they can't rise any further. This one got moved to the right. Probably that's a dilution issue because the replicates are really overlapping well. That's the PCR cycles. Here, probably these didn't have any DNA. So either they are negative controls or they're very low concentration samples. Exactly. So these are the data that you get, the primary data. In reality, this doesn't usually look that clean. So here you have a couple of reactions that failed completely. So you have to look at these curves never just take the table out of the machine, look at the curves and exclude those from analysis. Here, the threshold line, for my taste, is a bit high because it's like there's a sample here at the back that actually I would like to still see, but we could also put it a bit lower. The threshold line has to be in the area that is linear. So the threshold line cuts the linear range. If the curve starts to go towards the plateau and you put it here, that's wrong. So you really look for, you can usually manually set this in a given run, and you look for the place where they are all linear. And these look like standards because they are evenly spaced and the replicates overlap. And these are probably samples with very low concentrations, which is usually the case from environmental samples. So how do these then become this data? How are they transformed into the standard curve? Basically, what you do is you take these points where the threshold line cuts the curves, and you plot these against the known DNA concentration of your standards. And here it says DNA concentration that's overlapped by the line. And this gives you then a fairly, in this case, a fairly linear relation. More DNA. More DNA means more fluorescence. And so the CT, the low CT values come from samples with high DNA concentration. The high CT values come from samples with low DNA concentration. So basically the standard curve is CT against DNA concentration. Okay, and now you repeat this a lot. Very low dilutions, very high dilutions. You do this on different days. You do repeated dilutions. You alter primer concentrations, the primer ratios. You add anti-inhibitory agents. This is the place and time when you optimize your assay, in and out for weeks. Okay, and then you have some of these runs that look like we looked before. Some of them that like taper off. Some of them you catch a bit more like this, and in the end, you will end up with something like this. From many runs, you'll have an overlap of something that looks like this. You can see this is from a student who did 22 runs on different days on different machines. And from this, you can start to play around a bit. Exactly. Because the curve, these curves will shift, or also the standard curve will shift depending on the sensitivity of the assay. So if your assay gets more sensitive because you did something, then your amplification curve will shift as a whole to the left or the right, but it will stay in the same orientation. If you do something about the efficiency of your assay, it will change the angle, because it means that in every cycle there is less template, less new product, or in every cycle there is more new product. And this is, for example, something you see when you have inhibition in your sample. The curves will go flat, and if you then alleviate that inhibition through purifications, the curves will get back up. The effect on the CT is very similar. The CT crossing point that shifts, but you have to understand why it shifts to really know what you're doing. Exactly. So know your curves. Okay, so now we have the standard curve. This is the linear dynamic range. This is the range where every increase in DNA concentration leads to a decrease in CT. And again, the same, it's a correlation, right? It's a linear correlation. Where this stops is the limit of quantification. That means below this DNA concentration, you can still say you have something, but you can't anymore say how much you have. And then usually you have a limit of detection where you don't get any points up here anymore, and they all drop to no reaction, no amplification. And here, this looks a bit unusual because they stay up here. And we actually realized that there was a problem with this assay because these blue ones are negative controls. They shouldn't give any CT value, but they do, which means that in this range up here, in this case, we cannot trust our assay anymore at all because the negative controls also react. It was a big bummer because, and that's a known problem for this particular assay. It's a bummer because a lot of samples are in the same range. So we have no clue whether these samples are actually positive or negative. The good part about the linear dynamic range is once you know that, in every run, you will include just a few dilutions that are within the linear dynamic range, and then you can quantify within that run. Okay, just 3 to 4 samples of known concentration. After a few months, you know how reliable your pipetting is, you know how many replicates of this you need, and at some point, usually that's your permanent internal control. Okay, standard curve. So we have this LOD, LOQ, linear dynamic range. These are important, and sensitivity versus efficiency of the reaction. Know these if you do a qPCR. Well, and then we come to the qPCR itself. This you will see in the afternoon. But there's just one thing I would like to point you towards, and that's the MIKE, however you pronounce them, guidelines. They were published in 2009, and it's something like a guideline for which aspects of your assay you should publish and record. And this includes stuff like the proposal of consistent terms. We all have heard about qPCR or RT-PCR or RT-qPCR, like when to use which term. Actually, what they propose is to only use RT-qPCR for RNA, because RT, some people use it for reverse transcription and others for real time. And Q is enough for the real time, quantitative real time, and the RT only use when you start from RNA. Linear dynamic range is one term they propose, and sensitivity, analytical sensitivity, which would be what I introduced as LOD. I brought this article, maybe we can copy it afterwards for everybody who's interested, or you can just read it. And they say people usually under-report on their qPCR assays. Many papers with qPCR do not report the data, the software used to analyze the data, the decisions made, outliers that they threw out. They don't tell a lot about the primer characteristics and so on. So it might be a bit over the top what is proposed here, but it's a good start to make conscious decisions about what you do and what you don't do. So take homes. Lunch is soon. Work with a standard, a tractable standard, not some old positive sample, but with something you know what's in there and how much is in there. If you adopt the protocol, which is the most common scenario, test it, evaluate it, be in charge of that things. Even moving a test to a different machine can change the sensitivity, for example. So moving it from one lab to the other, of course, changes aspect of it. You have to reevaluate it and plan the time, right? I mean, if your supervisor tells you set this up, then we get the samples in 2 weeks and you can test them. Tell them no, give me 2 months. And yeah, again, if you order a new supplier, a new reagent, you have to, not with, if you reorder primer from the same company, no. But if there's something, for example, if a new person in the same lab starts the assay of somebody else, reevaluate this. In your hands, it might be a bit different. Okay, and with this, I'm at the end. This is the Phoebe team. We actually work on a wide range of things that might be interesting to you. Crayfish plague is one thing, aquaculture microbiomes is another one. We work on eDNA a lot, so detecting these things from flowing freshwater systems, on how to optimize this, and also, for example, how to statistically evaluate how good our detection is. And we actually have, you showed this script, the COVID tests. We're actually working on this for some fish parasites, not for zoosporic parasites at the moment, but here we try to find ways that people in the field can do it at a single temperature and so on. Amoeba and accredited diagnostic center.